Bowman. I am faculty in the Department of Neurology, and I lead our clinical trials and brain nutrition laboratory at the McCance Center for Brain Health. This is the McCance Center for Brain Health Seminar Series, and it's my pleasure to introduce Dr. Raja Bhattacharya, um, who is an assistant professor of neurology here at MGH and Harvard Medical School. He's also a cell biologist who studies the cellular and molecular mechanisms of Alzheimer's disease pathology. He received his PhD from Jadavpur University, Bose Institute in Calcutta, India. His doctoral thesis included studying fatty acid acetylation in the erythrocyte membrane, protein 4.2, and chronic myeloid leukemia. In his postdoctoral research, he uncovered aspects of fatty acid acetylation, specifically palmitylation, in regulating subcellular localization and functions of oncogenic genes, proteins, namely GALFA 12, 13, and Rho-RAC GDPases. Um, his current research is focused on palmitylation APP, how it's stabilized and prepared for cleavage by beta secretase and special lipid rafts within the neuron, which he has coined as mitochondria-associated ER membranes or MAMs. So today his presentation will focus on just that, targeting MAMs, stability, and endosomal abnormalities in neurons to prevent early stage AV pathogenesis. Dr. Bajra, welcome.
Thank you, Gene. Thank you for the introduction. Uh, and thank you all for attending. So let's go straight to, I have nothing to disclose. Um, so as you all know that Alzheimer's disease, which is a progressive neurodegenerative disease and characterized by amyloid plaques, neurofibrillary tangles, and neurodegeneration at the very end. Um, so neurotoxic A beta peptides are the building blocks of amyloid plaques and are released from neurons and form dimers, oligomers, protofibrils, fibrils, and then finally the plaques. The protofibrils can also be induces tau pathology. Uh, it's A beta is found long before the symptom appears, as you can see here in the brain, indicated by darking the red color along the progress of this stages from preclinical to clinical stages. And thus treating patients early is a key to successful therapy, which is supported by the recent clinical success of anti-amyloid immunotherapies, such as Lacumbi, and that slowed cognitive decline in patients with prodromal to mild AD by targeting and clearing A beta protofibril primarily in the brain and is now FDA approved for AD therapy. Despite the clinical successes, anti-A beta immunotherapy is less accessible owing to the associated safety concern and high cost of maintaining the patients to manage the side effects. Therefore, there is an urgent need to develop small molecule inhibitors targeting A beta pathology at the earliest stages. So I decided to divide my talk into two parts. First part will be to describe you the three aspects of Alzheimer's disease, and I'll describe our recently published studies demonstrating how these are interconnected. And these are ER mitochondria membrane contact sites. As you can see here, between mitochondria and ER, there is a contact region. I'll talk about that and tell you how these regulate several aspects of Alzheimer's disease. And then I'll talk about axonal A beta, which is a potential early event in AD and AD-related dementia. And then I'll talk about palmitylation, which is a fatty acid post-translational modification of proteins that targets proteins to lipid-rich environments such as lipid rafts. And the second part will be to discuss our another recently published paper and a paper that we have just submitted, to discuss about the role of two AD genes closely related, BIN1 and RIN3, and their effects on early pathology, which is RAF5 positive enlarged endosomal, enlarged endosome or endocytic dysfunctions. So let's start with part one. The first thing I want to discuss about APP, which is the amyloid beta precursor protein, which undergoes two kinds of processing. One is non-amyloidogenic processing, and another is the amyloidogenic processing. In the non-amyloidogenic processing, APP undergoes sequential cleavage at the plasma membrane by alpha secretase and gamma secretase complex to produces neuroprotective soluble APP alpha and P3, which is non-neurotoxic. On the other hand, amyloidogenic processing is by base and gamma secretase, producing amyloid beta and beta CTFs, both of which are neurotoxic, and this occurs primarily inside lipid raft domains. So as I told previously, the palmitoylation targets several proteins to lipid-based to lipid rafts. And it has been shown by Dr. Tinakaran's group and others that both base and gamma secretase are palmitoylated, and this palmitylation targets them into the lipid rafts. They also showed that knock-in of the palmitylation deficient base as well as the transgenic mice overexpressing palmitylation deficient gamma secretase subunits decreased both amyloid plaque burden and working memories in AD mice. However, the same mutants did not affect the enzymatic processing of APP in vitro and also in vivo. We have studied palmitylation in APP and found several papers have been submitted. We and others have found that APP is also palmitylated. But remember that APP palmitylation is very unique. It's a luminal palmitylation, which is one of the rarest among the transmembrane protein. And therefore, it is considered to be in the luminal lumen. And also, we have shown that it's palmitylated in the ER lumen. Approximately 10% of APP is palmitylated, but also it's important to note that with age, we have shown in mice that palmitylated APP levels increases. Therefore, it's a pathogenic target. It could be a pathogenic target. Most of these palmitylated APP are in the dimerized forms, and they are all are found in either in lipid rafts or in detergent-resistant membranes, which are also lipid-rich microdomains. Now, lipid rafts are mostly found in the plasma membrane, but also are found in many other intracellular membranes, such as MAM, which is an area in between mitochondria and ER, filled with hundreds of protein anchors holding these two organelles together. It has been shown, at least in many ER proteins, after synthesis in the ER, they get palmitylated and gets into these MAM areas. Two of these are most important to us because they are involved in calcium signaling. One is calnexin, another is TMX1, and they bidirectionally regulate MAM localized SARCA, which maintains the calcium homeostasis in a palmitylation dependent manner. And we call this membrane, although it is ideally a cytoplasmic area, because we can purify it biochemically as lipid rafts. As you can see here, we could isolate them by sequential centrifugation techniques and extraction, and you can see a pure MAM forming in this area, in this fraction, which can be detected with a MAM-specific protein VDAC1, and BIN1 is a soluble protein, is not present here, confirming that this is pure MAM. And we perform palmitylation assay in these MAM fractions to identify that APP is palmitoylated APP is primarily in the MAM fractions, but not in non-MAM fractions, whereas non-palmitoylated APP is not in the MAM fraction, suggesting that majority of palmitoylated APP is accumulated inside this ER lipid rafts. Here is a graphical representation of this Western blot. Now, recently, a group from Columbia University, led by Dr. Aria Gomez and Sean, has proposed or coined this hypothesis called MAM hypothesis, that the perturbation of MAM assembly and function is a key factor in AD pathogenesis. And they have shown that both sporadic as well as familial AD with various FAD mutations has shown perturbed MAMs across the disease progression, meaning that it has, because they saw this both in fibroblasts as well as in neurons. The perturbation of MAMs has many effects, three of which I have described here. One is calcium transport defects. It leads to mitochondrial dysfunction, and then it also leads to neurotoxic A beta increase, all of which, as we all know, leads to Alzheimer's disease. Now, two of these I described here. One is the calcium transport, which is regulated by this protein called sigma-1 receptor in the ER membrane. That controls this tripartite complex, IP3R3 in the ER, VDAC in the outer membrane of mitochondria, and GRP75 to shuttle calcium signals across ER and mitochondria, which is very important in maintaining neuronal plasticity. And our interest is because all base APP gamma secretase, including the beta CTF, and also with some question mark A beta, has been identified, has been isolated in MAM fractions, both in vitro and in vivo. A more recent study using a pharmacological modulators of MAMs called NE100 were able to reduce the MAM mitochondria forming MAMs significantly in both soma and axon. And a sigma-1 receptor knockout mice also showed a significant reduction in the MAM forming mitochondria, particularly in motor neurons. So we tested these two in our system and observed that the treatment with sigma-1 receptor, we call it agonist and antagonist also, although I do not want to call them these for reasons that I'll describe later. But no matter what, this PRE, any treatment that reduced the half-life of palmitoylated APP, as you can see here in a cyclohexamide chase assay, without affecting the stability of non-palmitoylated APP. We also showed that NE100 that destabilized APP also reduced the cell surface palmitoylated APP without affecting total APP. Now, sigma-1 receptor is becoming increasingly becoming an important therapeutic target for several neurological disorders, such as ALS, Parkinson's disease, Alzheimer's disease, and Huntington disease. These are type 2 transmembrane protein found in the ER and in MAM. And this is an atypical receptor. This is why I resist to call their agonist and antagonist as agonist and antagonist, because these do not have any second messenger system. Therefore, I would like to call them sigma-1 receptor modulators or ligands. And others agree to that too. And they are ligand-operated chaperones. They regulate calcium transport, as I showed. And several of these sigma-1 ligands or modulators with favorable pharmacological profiles are anti-amnestic. two of which are highlighted here because one of them is in phase 3 trial that targets both sigma-1 receptor and the muscarinic acid receptor, and these are small molecule therapy. And then rivastigmine, which is interesting sigma-1 receptor ligand that lowered A beta level and increased soluble S-APP alpha level, which is a protective product of APP. And I will talk about that again later. So in addition to regulating the ER and mitochondria contacts, ER mitochondria contacts, the most interesting aspect of it is its diversity in its both the structure as well as function. Structure, as you can see here in just one figure, you can see that there is a contact area between rough ER and smooth ER. These are the two main components of ER, forming two different kinds of thickness. And it has been calculated that the thickness of ER and mitochondria contacts or MAMs ranges between 6 to 80 nanometers. And these are not just the structural differences, they're also functional. Because a close collaborator of ours from Philadelphia, Dr. Heisnowski, has shown that by changing the gap width between ER and mitochondria, you can actually modulate the calcium signaling transport along ER and mitochondria. But it is not one directional. As you can see here, the thickness, when the thickness was about 35 nanometers, it did not transport calcium. At the same thing, when it was tightened, although there is a threshold where it does a better job. Therefore, it gives us an idea that the gap width might have a threshold that regulates functions. And it has also been shown that this threshold or this contacts regulate apoptosis, particularly in presence of pro-apoptotic protein BAC. And the cytosolic calcium levels remained unaffected by tightening or loosening the MAM gap width, which we already described before. Now, to understand the effect of sigma-1 receptor and MAM in AD, we took advantage of this 3D neural culture model called AD in a dish, developed by Dr. Kim and Dr. Choi, our very close collaborators with Dr. Tanzi. And these are basically a 3D neural model that produces soluble A beta in the conditioned media, and also A beta aggregates and plaques if you differentiate them long enough in this matrigel 3D matrix. Here is an example of amyloid beta in this culture, and here is an example by Sang Su's paper showing that you can also detect A beta. We used this clone 3C1 for our studies mainly, but we are using others too. And so we also detected, just like before, we also detected this rough and smooth ER forming visible, tight, and long or loose MAMs that we can count based on the electron density between these two organelles. And we decided to test our hypothesis in neurons because neurons are unique, where this smooth ER and rough ER are compartmentalized. It's not in any other cells. Therefore, it's very important that we use cells like neurons to study the effect of this gap width effect on amyloid hypothesis, on amyloid pathology. In addition to that, this system is also, we were able to we are able to develop a microfluidic chamber system where we could differentiate these cells, these neuronal cells, and can collect media from the axonal and from the somal areas, and can detect A beta production in both separately, which were not been able to, which were one of the hindrance to study axonal A beta. All the axonal A beta are shown to be one of the early stages of the disease. And for this, we must acknowledge Mehdi and Deepak for giving me synthetic A beta, and Mehdi for generating these microfluidic chambers. Now, when we knocked down sigma-1 receptor in the system and counted the ER and mitochondria contacts based on the electron density areas between these two, we observed a significant reduction in the tight MEMS, as you can see here between these two, and opposite effect on the loose MEMS. With sigma-1 receptor. And then also the sigma-1 receptor knockdown reduced the A beta 40-40 ratio, suggesting for the first time that sigma-1 receptor knockdown, that sigma-1 receptor upregulated amyloid pathology by modulating the levels of tight and loose MEMS, or tight or loose MEMS. Here is a quantitation technique, how we quantitated them. And this figure shows that the thickness, when they were bigger than 80 nanometers, we did not count them, and we counted them as non-MEMS. Now, to further test whether the thickness of ER mitochondria does indeed affect amyloid pathology, we used stabilizing stable molecular stabilizer that we obtained from our Philadelphia collaborator, Dr. Hajnowski, who used this for for his studies. And these are expression plasmids containing mitochondria targeted signal and ER targeted signal attached with different linkers. 1x is the shortest, the 9x is the medium, we could say the average MEMS, which is about 25 nanometers, and 18x is loose MEMS, which is about 40 nanometers. So this is 6 nanometers, 25 nanometers, and 80 nanometers. We used a mitochondria without ER as our control. And then we tested their effect in A beta by introducing these and then enriching these cells, these RAN cells, and developed in 3D, collected the media and measured A beta. Here is the expression levels of all these mutants. And as you see here, 1x showed a robust increase in both A beta 40 and 42 levels, suggesting, again, for the first time, that tightening of the MAM affect A beta in a negative way, meaning more A beta is produced or A beta is enhanced in this already AD neurons. So next we addressed whether these tightness affect the distribution of these MAMs in neurons. For this, we collected the desomatized axons, which is purified axons generated by differentiating these neurons on a porous membrane. And then we collected them, ran Western blot, and as you can see here, that we tested their purity by looking at the nuclear signaling, nuclear proteins such as lamin B and KDM1, which did not show anything in this desomatized axons, suggesting pure formation of axons. And then we tested the results, tested the expression levels of all these RFP tagged stabilizers, and see that we we could not detect any of this 18X, which is the thicker or the loose MAMs in these desomatized axon, again suggesting that axonal distribution of MAMs contributes to amyloid pathology. So, so, so we looked for the mechanism. How is this possible? Uh, Again, our collaborator has done a series of experiments to show that increasing the gap width shortens the length, while decreasing the gap width increases the length. And we hypothesized that increasing the length with shortened MAM will reduce the surface area of mitochondria and therefore will perturb its interaction with microtubule and will have effect on the mitochondrial transport rate, which is very important to maintain, the neuronal plasticity. And so we tested these effect on mitochondria trafficking. And as you can see here, 1X, which is the tight one, showed significantly reduced level of transport. This is live cells imaging and the chymography. And I don't know whether you can see the transport, but we could measure it. And we used a well-established analysis tool developed by our collaborator, Dr. Selina Lomio from Tufts University. And as you can see here, that 1X reduced both this average speed as well as the overall movement of mitochondria, whereas the 9X did not. These were replicated in naive cells, suggesting that the effects are not A beta or APP dependent, but only due to the contact between these two organelles. Here is a tabular form which we developed in order to provide a blueprint to determine the efficacies of MAM modulating small molecule ligands, such as the sigma-1 receptor ligands, in changing the MAM gap width and affecting the amyloid beta pathology. For example, if you find average speed, if you find the average speed of mitochondria as 0.3, you can suggest that they would be A beta producing MAMs, whereas mitochondria average speed of mitochondria in the axons as 6 will suggest a reduction or the lowering of amyloid pathology. So next, we decided to test this in using the sigma-1 receptor ligand. We used PRE and NE, isolated MAMs after treatment, and you see the MAM fractions increased, the MAM levels increased by fourfold with PRE treatment and decreased by 50% in any treatment. However, we haven't detected any changes in the APP processing, either alpha or beta processing. Since we are interested in the axonal tight MAMs, we isolated axons, again, thanks to Mehdi for giving us this unique 3D dual chambers, microfluidic chambers. And we could do an axonopathy and collect these axons. And here, with a PRE treatment, we see a significant increase in the MAM proteins. We used 3 different MAM proteins: IP3R3, ACAT1, and VDAC1. They all increased with PRE, while they decreased with any treatment. Next, we made another state-of-the-art technique to isolate pure A beta from the axonal chamber, and then calculated the A beta molecules per soma and axon to show that any treatment dramatically reduced the A beta levels only in the axons, but not in soma, whereas PRE did the opposite as expected. By extending this sigma-1 receptor studies, we also used rivastigmine, which I was talking about in the beginning, which is an anti-amnestic drug used in AD treatment and has been shown to reduce A beta in culture model, as well as its increase of SFPB alpha in this rat cortical cultures. So we treated our 3D neural culture model with rivastigmine and observed somewhat dose-dependent reduction, which reached significance at around 50 micromolar without affecting the MAM proteins. And these are the loading control. So we see that sigma-1 receptor ligand rivastigmine reduced A beta, but did it do it by changing the ER mitochondria contacts? For this, we developed another a FRET-based assay where we used this ER-targeted CFP, mito-targeted YFP, linked with this self-cleaving TAB2, which upon cleavage produces equimolar levels of these two, and then produces FRET when they come closer, which is when they come when they bind to the ER and mitochondria that are tightly bound by MAMs. So we performed this assay in vitro. We used Neuro2 cells in this case, and as you can see here, this is a CFP expression, YFP expression merged, and we also used a GFP labeling to confirm that these are all expressing where they should be. And then we treated these cells with rivastigmine. They did not change the molar ratio between these two biosensors, where, however, the FRET ratio dramatically decreased upon rivastigmine treatment, and also A beta 40 reduced, again confirming gap with dependent A beta production. Here is the tabular form suggesting that FRET ratio changing from 1.16 to 1, or FLIM, which I did not show here, which increased from 1500 to 1900. As you will know that FRET and FLIM are the opposite. And then you can calculate the A beta 40 ratio, but these are all done in Neuro2 cells. So what we have so far detected is that the impact of sigma-1 receptor ligands on A beta levels in 3D neuronal model is that PRE upregulated axonal MAMs and exacerbated neurotoxic axonal A beta levels in our 3D system. Whereas NE100 and rivastigmine downregulated axonal MAMs and reduced A beta level respectively by ameliorated neurotoxic axonal A beta and reduced total A beta respectively by loosening or by downregulating axonal MAMs. Now, as I said, that I do not want to consider sigma-1 receptor agonist and antagonist because of their atypical nature of sigma-1 receptor. If we look at rivastigmine, it has an affinity not just for sigma-1 receptor, but also for sigma-2 receptor, whereas both these have strong affinity towards sigma-1 receptor, but no visible affinity towards sigma-2 receptor. However, it is important to understand that sigma-1 and sigma-2 receptor, in spite of their similar nomenclature, are not splice variants. They have only the overlapping pharmacology, and they are implicated in neuronal disorders. They are both from two different chromosomes. Nowadays, sigma-2 receptors are called TMEM97, which is from the chromosome 17. Sigma-1 receptor is a type 2 transmembrane protein in the ER and MAM, whereas sigma-2 is a type 4 transmembrane protein. So there is no similarity, except that pharmacologically they are very similar. So we tested the effect of sigma-1 receptor knockdown. I don't have the time to show you that, and we are quite confident that sigma-2 receptor is not the effector of ER mitochondria contact changes. To test the physiological relevance of MAM gap width in AD, we performed electron microscopy of mouse optic nerve, which serves as a reliable model of pure axons because about 90% of the axons arising from the nasal retina project through the contralateral optic tracts to the contralateral brain, and therefore it's a physiological representation of pure axons. And most importantly, A beta accumulation in the retina precedes the accumulation in the brain. So we still don't know why we observed a significant increase in the percent mitochondria forming MAMs in 5x FAD compared to the wild type, but we also see very similar effect in human retina that we have obtained from a collaborator of ours. I have no time to show that, but we are studying the protein composition in these MAMs isolated from these optic nerves, both mouse and human. So to summarize the part one is that first we showed that palmitylation targets PAL-APP to MAMs, and possibly there is a non-canonical secretory pathway for PAL-APP that goes to the plasma, that takes it to the plasma membrane by bypassing the classical ER Golgi plasma membrane secretory pathway. And it'll be very interesting to determine how this non-canonical pathway works, and maybe the mechanism will provide enough evidence to study this pathway for better therapeutics. The second is that the MAM gap width stability is a key factor in initiating amyloid pathology. And we have shown at least two FDA-approved and pharmacologically safe sigma-1 receptor targeting MAM gap width might lower A beta as an alternative to A beta immunotherapy. I have added calcium here because the calcium, as I showed, transport has been affected by this gap width, which is not surprising because of the diffusion chemistry, but A beta is a much larger peptide compared to calcium molecules, of course. So therefore, it'll be very interesting to know how A beta in this area contributes to amyloid pathology. So next, part two. Here, we would like to describe the role of two AD-related proteins, sigma-1 receptor, AD-related proteins, BIN1 and RIN3, in modulating this endocytic dysfunction and the endosomal abnormalities such as RAB5 positive enlarged endosome that is shown by Quart et al. And this enlarged endosomes are considered to be an early pathological events of AD and AD-related diseases. Here is the quantitation of different APP mutations, FAD mutations in both APP and PSN1, as well as together showing significantly increased levels of this endosomal pathology in HIPSC-derived AD neurons. More recently, it has been proposed by Ralph Nixon's group that they use this directly overactivating RAB5 pathological mouse model and showed that they mimics the AD-like endosomal dysfunction, as you can see here, endosomal RAB5 positive endosome enlargement, which is here quantitated. So the puncta size were quantitated and also quantitated per cell, showing that the pathological RAB5 increased this RAF5 positive enlarged endosome. And they also showed that these mice have synaptic function deficits. They also show tauopathy, and they resulted in memory deficits. Therefore, this RAF5 positive endosomes are not only developing this early stage pathology, but also may be involved in much later stages. So they suggested that RAV5 is a potential therapeutic target for AD or AD-related dementia. However, if you know that RAV5 is a molecular switch that changes between inactive GDP bound to active GTP bound form and is tightly regulated by guanine nucleotide exchange factors and GDP dissociation inhibitors to maintain the endocytosis of cellular endocytosis. Therefore, targeting RAB may not be a good method to lower RAB-mediated endosomal pathology. However, there is this protein called RIN3, RAS and RAB interacting protein, which acts as an AD risk, which is an AD risk gene, but it's not understood how they affect the amyloid, how they affect AD. But several common variants of RIN3 have been identified as major genetic risk factors for AD and are associated with tau pathology and intracellular A beta accumulation. RIN3, as I said, acts as an GEF by transforming RAB5 family of proteins, including 5 and 31, but not RAB7. And RAB7 is involved in lysosomal development. RIN3 is a direct binding partner of BIN1, which is a major AD susceptibility gene product with an effect size on risks second only to ApoE. So BIN1, and there is this BIN1 undergoes an extensive splicing leading to at least 12 isoforms, 1 to 12. Two of the isoforms are studied mostly because of their size differences and the addition of this clap domain in one of these isoforms, and they are found mostly in the neurons and astrocytes, whereas the forms that do not have the clap domains are found in oligodendrocytes. Neuronal BIN1 may have some effect on both tau pathology and base1 trafficking, whereas non-neuronal V9 is a key regulator of microglia activation and also plays a role in neonatal myelination. If you knock down BIN1, they're perinatally lethal due to cardiomyopathy, and therefore it has been extremely difficult to study the BIN1 effect in animal models. However, BIN1 and RIN3 form a tripartite complex with active RAF5. So recently, in an in vitro study, we reported that the neuronal BIN1 V1 negatively regulated APP endocytosis and A beta secretion upon translocating to RAB5 positive endosome. As you see here, this is V1, this is V9. V1 is on the RAB5 positive endosomes. And this is a co-localization study showing a significant decrease of co-localization. And we have done several other assays to prove this, to finally conclude that BIN1 V1 is usually in the closed formation, but when bound to RIN3, their clap domain gets exposed or opened, and therefore interacts with the clathrin-mediated pathway, which is how APP is also transported into the endosomes, and therefore perturb or delays APP endocytosis, whereas does not have any effect on base 1 endocytosis. So after this, we first tested if we knock out RIN3 in mouse model, and we, after knocking it out, we prepared a hemizygous for RIN3 knockout mice, which reduced AD-like pathology, AD-like endosomal pathology. As you can see here, that we counted the endosome size, and you see the enlarged endosome was significantly reduced in this RIN3 hemizygous mice. Although these are RAB5 active labeling, and we counted them, there was no significant effect. These mice are viable as we tested them, and the partial knockout significantly reduced the numbers of RAB5 positive enlarged endosomes without affecting total active RAB5 levels. And the partial knockdown of RIN3 significantly reduced the number of RAB5 enlarged endosomes in brain without affecting the levels of total or active RAB5. So we have just submitted this paper, and now, as you know, that the disease-associated rare variants or SNPs help uncover obscure pathological pathways because they are less constrained by natural selection and are more likely to disrupt protein functions or gene regulation than common variants. As recently, Dr. Tanzi and Dr. Prokopenko identified like 16 novel Alzheimer's disease loci using this multi-ancestry meta-analysis, and they helped and gave us this two rare SNPs in RIN3. These are rare variants of RIN3, and the family-based GWAS, from the family-based GWAS studies, their allele frequencies is 0.01 to 0.2, although the p-value is higher in this one case, but they are considered significant because of their tight linkage disequilibrium with each other. And also these are common SNPs found near RIN3, which are previously found by strong association with AD. These two SNPs also produces missense mutations in the PRD, which is the proline-rich domain of RIN3, which is considered to be the binding domain for BIN1. And in a first we tested the effect of BIN1 by knocking out BIN1 in neurons derived from HIPSCs. As you see, this was done by Anna. And you see there is no BIN1 expression. And with 6 days or 12 days differentiation, we start seeing neuronal form of BIN1, whereas before it was mainly non-neuronal, suggesting that this is neurons. These are neurons. And we performed an RAB activation assay to show that in absence of BIN1, the RAB was hyperactivated because there is no, in absence of BIN1, there's hyperactivated as was quantitated here, suggesting that in absence of BIN1, RIN3 overactivates RAB5, but when BIN1 is brought to the endosomes, it downregulates or it prevents RIN3 mediated RAB5 activation. We also counted the RAB5 positive endosomes in the neurons individually and locally. As you can see, we measured 0.1 to 0.5 microns endosomes as early endosomes, and large endosomes as 0.5 to 2 microns, and showed that enlarged endosomes were increased. The number of enlarged endosomes were increased in knockout neurons compared to the wild type. We also performed a bulk RNA sequencing to identify several AD-related as well as a novel gene that we'll be studying. A few of them are listed here. So all of these are involved in Rho and Rab GTPase activities, cholesterol transport, and then Wnt signaling pathway, as well as cadherin linked EMT pathways, Wnt beta signaling pathways, and cadherin signaling pathways. These are and I don't want to go into details about others that, so in summary, I want to suggest that so far we have shown that BIN1 knockout leads to hyperactivation of RAV5 in HIPSC-derived neurons. BIN1 knockout leads to endosomal enlargement in HIPSC-derived neurons. And I didn't have time to show you that we obtained two rare RIN3 missense mutations found to be overrepresented in AD case in late onset AD. And we introduced these mutations in HIPSC neurons and showed that these two RIN3 mutation residing in this BIN1 binding domain impairs binding to BIN1 and leads to hyperactivation of RAV5. I did not have the time to show this, but this is our ultimate goal to study the interaction between RIN3 and BIN1 in RAV5 overactivation. We also showed that RIN3 knockout decreased the levels of RAV5 positive enlarged endosome. So finally, I come to the acknowledgement. I must acknowledge Dr. Tanzi for giving me this opportunity to do this study from the beginning. He was supportive to this, and I should also acknowledge Dr. Kovacs. In her lab, I started this palmitylation studies. Jake has done a remarkable job. All the studies that I showed, majority of them are done by Jake. And Sehun, Mehdi, Du, and Dimitri, Louisa, they all contributed to this work. Anna has produced this knockout and edited HIPSCs. Sang Su gave me the expression plasmids of RIN3. And then, of course, Dr. Hajnowski from Philadelphia gave me those linkers. Masado helped me with Fred film assay, and Selena helped with the chymography assay, and supported by Cure Alzheimer and NIH. Thank you.
Thank you very much for that wonderful presentation. Let's see, we have a few minutes. We have 5 minutes for questions. You can post your questions in the chat or please raise your hand. I have a couple questions. Rudy, do you have a question you'd like to open up with?
Sure. Yeah, great job, Rajan, and very exciting about these new mutations we found that disrupt the RIN3 RAB5, the RIN3 BIN1 binding that upregulates RAB5. So in thinking about RAB5 and its effect on endosomes and this idea of endosomal enlargement as part of AD pathology, can you just explain maybe how does the field view endosomal enlargement relative to A beta deposition, tauopathy, neuroinflammation in the Alzheimer's pathological cascade?
Yeah, so that's exactly what this endosomal pathology leads to. Is usually called endolysosomal pathway that is activated in early on, and the lysosome to autophagy is another pathway that is activated, and therefore this enlargement of endosome might suggest activation of clearance of either. I don't know whether it is for A beta, but at least clearance of other proteins involved in APP processing by increasing their lysosomal degradation, then
I'm sorry, Roger, but I was asking about swollen endosomes and how they relate to AD pathology, not the role of endosomes, but how these swollen endosomes, how do they relate to amyloid and tau and other AD pathology?
Um, enlarged endosomes will have uh, I don't know exactly enlarged endosome, but enlarged endosome leading to lysosomal degradation is the only thing that has been quite aggressively studied. But we have to how this enlarged endosomes. I would suggest that the enlarged endosomes increases the APP endocytosis, and that's how they could affect the A beta production, either increasing the intracellular A beta levels, or by disrupting the APP A beta APP base pathology, APP base cleavage, by switching the APP from, say, alpha secretase cleavage at the cell's surface, and improve and directing it towards the base cleavage in the endosomes.
Thank you.
Hey, we have another question, Martin Zhang in the chat. Do you think AD-related MAM changes may be a compensatory mechanism in response to intracellular or medium extracellular A beta stress and toxicity? Then the second part is the S1R reduced A beta 42 to 40 ratios. Do you detect A beta from medium or the lysates? Thanks.
The second question first is that we detect A beta from both. The one that I showed is from the extracts. So we see both changes in both from the soluble as well as from the extracts. The first question is AD-related MAM changes may be compensatory to mechanism in response to interest. Yes. Stress, yes, of course. The ER stress is one of the major contributing factors for AD. Therefore, it is quite. I'm quite certain that the A beta generated in these, or particularly beta CTF, which has been shown to be the major factor in stabilizing these contacts. So they definitely would have stress and contributes to. I don't know that it's a compensatory pathway. I don't know why you're saying compensatory in what sense.
Maybe we can take that offline since we're down to just a minute here. We have one more question from Yu Yao Tian.
Hello, hello, Raj. Yeah, very impressive talk. And my question is, do you think the ring 3 beam1 interaction that's specific to neuron, or it also functional in the glial cell, like the astrocytes?
So the differences that I observed, at least in my in vitro studies, is that the interaction between the neuronal isoform is what is affecting the BIN1 mediated at least endocytosis as well as A beta APP processing. I did not see any effect of the non-neuronal isoforms, so I assume that it's most likely the neurons are the right cells where these interaction causes the defects.
I see. Thank you.
Okay, Raja, thank you so much for your presentation today. Very stimulating, interesting on these MAMs as a new mechanism and target in AD pathology prevention. Thank you all for attending. We've run short on time here, and we will be back in touch with our next presentation in a couple weeks. Until then, happy April. Thank you all.
Thank you.